Assalamualaikum and a very good morning to Dr. Aida and my fellow friends. Today, me and my partner Nur Akmal binti Abu Bakar from Group 2 will be presenting about what are the improvements in terms of technology that the company can make to advance further in the industry. Compare our suggestion with the company current technology. Lablink current technology is polymerase chain reaction (PCR). PCR has been used to run many kinds of tests, and we will explain more about it in the next few slides. So what is PCR, polymerase chain reaction? PCR is a method widely used to rapidly make millions to billions of copies of a specific DNA sample, which allows scientists to take a very small sample of DNA and amplify it to a large enough amount to study in detail. As we know, in this field, we have many types of PCR. So in this presentation, I will be focusing more on real-time PCR because Lablink use real-time PCR to detect COVID-19 virus. So what is RT-PCR? RT-PCR is a molecular biology technique used to monitor the amplification of the target DNA or RNA sequence during the PCR in real time. So what is the difference between PCR and real-time PCR? Conventional PCR can only be visualized by running amplified DNA samples through electrophoresis. Meanwhile, in real-time PCR, the amplified DNA is labeled usually with fluorescent dyes, enable the amplification of DNA visually by reading the fluorescence of the amplified DNA. And the second difference is that after the amplification, the PCR products or the amplicant are run on the agarose gel electrophoresis to detect the presence or absence of DNA amplification. Meanwhile, in the real-time PCR, the amplification during each PCR cycle is monitored in a real-time manner and only can be visualized by reading the fluorescence dye. As I mentioned earlier, in Lablink, RT-PCR has been used for COVID-19 detection. RT-PCR also has been used for Zika virus detection and meningitis viral pathogen detection. Before I go further, I will explain a bit about principle of real-time PCR. The process is monitored in real time. The reaction is placed into a real-time PCR machine that watches the reaction occur with a camera or detector. The amplification of DNA is linked to the generation of fluorescence, which can simply be detected with a camera during each PCR cycle. The principle of real-time PCR relies fully on fluorescence dye. Hence, as the number of gene copies increases during the reaction, so does the fluorescence, indicating the progress of the reaction, meaning that the amount of fluorescence released during the reaction is directly proportional to the amount of amplified DNA. For your information, there are four main steps involved in RT-PCR, which are denaturation, annealing, extension, and detection. But right now, since we focus on COVID-19 virus, as Lablink use RT-PCR to run COVID-19 virus detection, so we will be explaining on how does RT-PCR work with COVID-19 virus in Lablink company. So the first step is a sample is collected from the parts of the body where the COVID-19 virus gathers, such as from the nose, and then the sample is being treated with chemical solution to remove unwanted substances such as protein and fat, so that only the RNA is present in the sample. The RNA is then being reverse transcribed to DNA using reverse transcriptase. The second step is scientists then add additional short fragments of DNA that are complementary to specific parts of the transcribed viral DNA. When a virus is present in the sample, then this fragment will attach themselves to the target section of viral DNA. The third step is the mixture is then placed in an RT-PCR machine. The mixture will go through cycles in different temperatures to create new identical copies of target sections of viral DNA. The cycle will go through and repeated over and over again to continue copying the target section of viral DNA, and each cycle will double the previous number. The fourth step is as new copies of the viral DNA sections are built, the marker label attached to the DNA strand and then release a fluorescence dye which will be measured by the computer machine and will be presented in real time on the screen. The last step is the computer checks the amount of fluorescence in the sample after each cycle. When a certain level of fluorescence is surpassed, this confirms that the virus is present in the sample. This is the last slide for my part, which is the current technology that has been used in Lablink. The advantages and disadvantages of real-time PCR. The advantages of real-time PCR is they are cost-effective as they don't use much chemicals to run electrophoresis process. And second is time efficient because RT-PCR only took 30 minutes to 2 hours to give result. More sensitivity and specificity because whole process in the PCR cannot be influenced by the non-specific binding. Increased dynamic range of detection, and then lastly is no post-PCR processing. The disadvantages of RT-PCR is the kits are not available for all kinds of genes and disorders because the technical and standardized protocols involved are very limited. Cross-contamination between nucleic acid can occur as the nucleic acid that has been previously analyzed from the past experiments might be present in a large number in the lab, so they might have the possibility for the cross-contamination to occur.
So I will continue with the improvement that can be made by Lablink, which is next generation sequencing. NGS refers to technique in which millions of nucleotide sequence deciphered simultaneously, and also known as high throughput or massively parallel sequencing for DNA, RNA, and methylation sequencing that evolved from traditional fragment-based Sanger DNA sequencing technologies. NGS allows for rapidly sequence whole genome, deeply sequence target regions, utilize RNA sequencing to discover novel RNA variants and splice site, or to quantify mRNA for gene expression analysis, analyze epigenetic factors such as genome-wide DNA methylations and DNA protein interactions, sequence cancer sample to study rare somatic variants and tumor subclones, study human microbiome, and identify novel pathogens. NGS divided into two generations, which is second generation and third generation. Second generation requires short read sequencing method, and the sequence is less than 200 to 400 base pair. It's cheaper to produce, which is use less reagent and less cycle, and complete when have a reference genome. While the third generation is a long read sequencing method, and the sequence is more than 400 base pair. It provides more sequence that can be used to assemble genome or transcriptome. This is NGS workflow in clinical pathology. This shows that the standard workflow for NGS assay. There are numerous steps involved which require technical precision. The steps may vary depending on type of assay and information required. So after the data analysis, there are validation reactions and clinical decision steps. So the advantages of NGS is higher sensitivity to detect low frequency variants, fast turnaround time for high sample volumes, comprehensive genomic coverage, lower limit of detection, higher throughput with sample multiplexing, and ability to sequence hundreds to thousands of genes or genes region simultaneously. For the disadvantages of NGS, short reading sequence, the challenges in assembly of the reads, complicated data analysis, and large data storage required. So the application in NGS application, mutation detection in Mendelian disease, recognizing disease predispositions and protective factor, pharmacogenomic, improved diagnosis and treatment of cancer, epigenetic, in identification of structural variants, and sequencing cell-free DNA fragment. The comparison between NGS and PCR is, for the benefit PCR, it has a familiar workflow and capital equipment already placed in most lab. For NGS, it is higher discovery power and higher sample throughput. For the challenges for PCR, it can only interrogate limited set of variants, virtually no discovery power, and low scalability. For the NGS, less cost effective for sequencing low number of targets between 1 to 20 targets, and time consuming for sequencing low numbers of targets.